Innovative Young Biotechnologist Award, Postdoctoral Fellowship from Tsukuba, Japan. And now she is continuing as Assistant Professor at INQUEST Institute of Information Technology.
And thank you for the kind words in the introduction, sir. So, Dr. Soma has already made my job much easier. So, whatever she explained in detail is the initial part of my slides. So, you will see a lot of repetition from what she has already talked about. But yes, what I will be talking about today mainly deals with the anti-COVID potential of ashwagandha and honeybee propolis. And along with the NCU analysis that I'll be showing you, we have moved a step ahead and did some in vitro validation. And we collaborated with RCB to do some other viral-based assays. Okay? Just to start with, so just very general introduction. As we all know, we do not really need an introduction to COVID-19 situation. We all have experienced it in some or the other way during the last 3 years. But this figure shows you the most recent statistics. So the problem is not yet solved. So you can see over here, this is the number of cases that are reported in the last 24 hours. So this data is for October 6th. And this is the number of infections that were observed in the last 24 hours. And this is the total number of cases that have been reported so far across the globe. And it has already infected more than 215 countries. Right? And this is a brief introduction to the structure and genome of SARS-CoV-2. So it is mainly 29.9 kb in length, the genome. It is a single-stranded positive sense RNA virus, right? So mainly the two-third part of the genome, it encodes for 2 polypeptides, which encode for a total of 16 non-structural proteins. And these are some of the important proteins which are involved in the replication and the progressive infectivity of the virus. And this 1/3 part of the genome encodes for the structural proteins, which help in the making of the viral particles. And this is how the virus actually looks like. So Dr. Soma has already talked about these different components, right? She mainly focused on these 5 proteins. So we have this nucleocapsid, membrane, envelope. All these form the structural component of the virus. Okay? This is also something that she has already shown. So this is a schematic representation of how this SARS-CoV-2 infects. So I would like to reiterate on this part. So for the virus to enter the host cell, these 2 host receptors are important. She mentioned about this ACE2 receptor, and there is one more receptor involved, that is TMPRSS2, right? So this angiotensin converting enzyme 2, this is the first enzyme with which the virus particle interacts by the S1 subunit, and then this TMPRSS2, it facilitates the proteolytic cleavage of this spike protein, which then further facilitates its fusion with the membrane, and it is endocytosed into the cell. Then this genetic component of the virus particle, it is released inside the cell, and using the host machinery, it encodes for these polypeptides that I was talking about, encoded by the 2/3 part of the genome. And there are certain proteolytic enzymes which are also encoded during this process, and they cleave these polypeptides to make the functional proteins. And during this part, this enzyme is formed, RNA-dependent RNA polymerase, which further helps in the replication and the transcription of the virus. So this is the general process, and after this, the virus particles, they keep on growing and infecting the further cells. So these are the symptoms which are associated with this COVID-19 infection, right? And these, during that time of SARS-CoV-2 infection, so these were some of the drugs which were repurposed, and they were shown to be effective against these molecular targets in the entire replication cycle of the SARS-CoV-2. Okay? So for the work that I'll be talking about today, I'll be mainly focusing on these 2 as molecular targets for ashwagandha and honeybee propolis based bioactive compounds. And along with that, I'll also be talking about this 3CL-Pro, which is also known as the main protease of the SARS-CoV-2. Right? So as I said, we are targeting TMPRSS2, which is transmembrane protease serine 2, then AC2, this we have already talked about, and the main protease or the 3CL-Pro of the SARS-CoV-2. Right? So why ashwagandha and honeybee propolis? So this is the part of the work that I'm presenting today. This was done as during my postdoctoral fellowship at AIST in Japan. So the lab already works on these natural compounds, and we are looking into the anti-cancer potential of these natural compounds. But these natural compounds, they are also known for their antiviral activity. So there are many reports in literature that talk about the bioactives from this ashwagandha and they have been shown to be effective against H1N1 virus, influenza, herpes simplex virus and even HIV-1 virus. So this was one of the important plants that could be looked into for some repurposing work, right? So these are the therapeutic activities which are associated with the ashwagandha bioactives, and these are the two bioactive compounds which are most abundantly present in these plants, right? So Bidopterin A and Bidopterin. So not only these two main bioactive compounds, but there are many other different regularites which can be used from ashwagandha for various therapeutic activities. So for our study, we looked into these small molecules which are extracted from ashwagandha and are abundantly present in their extracts. Along with that, this is another small molecule that has been used. I will tell you the use of this in the coming slides. Okay. So first of all, we wanted to see for the preventative measures of these ashwagandha compounds. So for preventative measures, we wanted to see if it can somehow help in reducing the interaction between the spike protein and the host receptors, TMPRSS2 and ACE2. Right? So to start with, we started with TMPRSS2. So the TMPRSS2 structure is not available in PBB. So the first thing was to model the protein so that we can look into some structural aspects of interaction of these small molecules with the protein. And the model structure is also available in the Swiss model repository. So it is modeled using the template serine protease hexane of human. And the identity between the human and like the template and this TMPRSS molecule is 33.82%. And using the structural studies and some reported literature, it was found that this domain, which is actually involved in the priming process of the spike protein, it is highly conserved. And these are the three catalytic residues which perform this priming or cleavage process. Right? So this histidine 296, serine 441 and aspartate 345. Right? Now when we are doing docking studies, now the theory behind all these experiments have already been explained by Dr. Soma, so I will not get into the detail. I will just touch upon those and I will come to the results. So when we are doing docking experiments, the docking score that we get, they are not absolute scores. So they are always like we can use them in relative to some known molecules. So this chemostat mesylate is one of the known molecules which can inhibit TMPRSS2. We have experimental evidences for this and a lot of experimental reports are also available. So this molecule, it is clinically approved and it is also extensively used for the treatment of oral cancer and some chronic pancreatic disorders. And recent reports, like the reports during this COVID time, they showed its importance for controlling this TMPRSS2 as well. So the first, and its molecular mechanism of inhibition is that it interacts with these three catalytic residues, making them unavailable for interaction with the spike protein. Right? So the first thing was to dock this chemostat mesylate into the same pocket and see what is the interaction energy or binding affinity of this particular small molecule with our target of interest. So chemostat mesylate docking, and we got a docking score of minus 5.9 kilocalorie. So this acted as our reference or control molecule for the in silico analysis. Right? Similar to the in vitro experiments, we do carry some control or reference molecules for the in silico analysis also. So this is one of the known inhibitors of TMPRSS2. Similar to that, we used these different vidarolites which I showed you in the previous slide, and we saw whether they can mimic the same kind of binding behavior against TMPRSS2 or not. Right? So if you look at these figures, these yellow dotted lines, they show you the hydrogen bonds, and apart from these major hydrogen bonds, there were a lot many other residues which were helping this ligand to stably interact with this pocket with the help of hydrophobic and wonderful interactions. They are not shown over here because they are lot in number and the image would not have been clear. So for Bidopterin A, the docking score was minus 5.6 kilocalorie per mole, and you can see that this molecule, because I am not showing you the entire protein as such, so this is this zoomed in view. So that these all molecules are interacting in the catalytic pocket that I showed you. So that is evident by this serine 441. So you will be able to see this in all the figures. So this molecule was interacting in the way similar to the chemostat mesylate that I showed you in the previous slide and is our control for these experiments. Right? So Bidopterin A, then Vidinone over here, docking scores are listed below the images. Similarly, within a light A and within a site 5, right? Here you have within a site 4 and methoxy with the pyrid A, right? Then we have within a light B and 2-deoxy, sorry, I cannot read this thing, 2-deoxy with a sorry for the name. So all the docking scores you can see over here. So as Dr. Soma mentioned, she said that -5 is the relative scale upon which you can say the docking score is quite good. But yes, there are certain molecules which we have seen in our personal experience also, that even -2 docking score, but when it is in relative in reference to some known molecules, they do give very good stable binding and they can also be translated in vitro experiments. So I would not say -5 is the threshold, but yes, you can always check in relation to the known molecular inhibitors. Right? So here some of the molecules they showed better binding affinity in comparison to this chemostat, but other molecules were also comparably similar. And the molecular mechanism of interaction was very similar to the non-inhibitor, and mostly all the molecules they were stably interacting over there with the help of hydrogen bonds, right? But yes, as she mentioned this also, that docking it generally represents only a static picture. So you just have a receptor molecule, you have the cavity in whatever form you are, like I'm using a model structure. So the cavity that I have just tried to fit that small ligand into the cavity in the best possible manner, right? But we know that in our body everything is in dynamic motion. So that is what these MD simulations generally reflect. So what we generally do, we take these dot complexes, we put them in a box of solvent which generally consists of water, and we try to see what happens to this over a given period of time, right? So as she mentioned, these molecules have been simulated for 100 nanosecond over here, because within these 100 nanosecond we could see that the ligand is stably interacting with it, right? So she discussed about the importance of RMSD, RMSF, So here what we are trying to look at, after the docking of the ligand with the protein receptor, whether there are some major structural distortions in the molecule or not, or is our new complexes showing a similar behavior to that of chemostat or not, right? So as we can see over here, the RMSD, it deviates a lot from its initial dock pose, but during the time of simulation, it remains almost the same, right? So it takes some time to adapt to its, most optimal conformation, but after that, it stays in the same state. So this overall represents the stability of the protein ligand complex. But over here, this is only for the ligands. So it is possible that the ligand, like we generally see that sometimes if the binding is not that appropriate, the ligand leaves its original place, it gets and unspecifically goes and binds to some outer surface of the protein, and it stays there stably for the entire simulation. But here the stable RMSD for the ligands alone, they were also telling us that the ligands were stably bound in the initial place where they were docked, right? And again, the RMSF, it did not show any deviation in comparison to the complex with the non-inhibitor, that is chemostat, and hence the overall complexes were very similar to what we were looking for, right? So this is what we initially analyzed from the molecular dynamic simulations. And yes, the interaction profiles that she was showing. So over here, the green represents the hydrogen bonds, which define the specificity of small molecules towards the receptors. Then the hydrophobic interactions, yes, they are also very important component when we are talking about interactions or stability of these small molecules and protein complexes. Some ionic interactions, but in most of the complexes, like in all these complexes, we will not be able to see these interactions, and some water-mediated interactions. As I said, simulations are carried out in water boxes, right? So this is the profile for chemostat mesylate, right? And this shows, like I said, the simulations were carried out for 100 nanosecond, right? So if we consider the entire time frame as one fraction, so this shows you the fraction of time for which a particular interaction with a residue was important. So here the residues are not very clear, we need not bother about it for now. But yes, the interactions, these were the interactions, and this is the fraction of time. So though the fraction of time maximum should be 1, but you can see up to 2 over here, because one residue could be involved in more than one hydrogen bond formation, right? So similar to this, if you look at the interaction profiles of the vithanolides of our interest, so the interaction was always better than that in the chemostat mesylate, or it was comparable to that. So yes, whatever we are thinking of right till now, that these molecules can show similar behavior in comparison to chemostat is still with us, right? So, after this, now we were like confident to take these to the in vitro analysis. So the first thing was to look at different cell lines. Now these molecules, because like as I said, we mainly work with antiviral potential, like looking into the antiviral potential of the nutraceuticals, right? So we looked into the expression of our molecular target of interest, that is TMPRSS2, into different cancer cell lines, and it shows the cell lines which express TMPRSS2 from moderate to high levels. And I'm showing you, like, these are the representative figures only, but we have done the study in multiple cell lines to be sure of what we are looking at, right? So these are the different vidanolites, like the numbers are representing vidanolites, what dosages are being used over here, and this figure shows you the effect of these vidanolites on the mRNA level of these different, like in TTN cell line, the mRNA level of TMPRSS2, what has happened to that upon treatment with these different vidanolites. So as you can see, most of the vidanolites could downregulate the mRNA levels of TMPRSS2, right? And this is in TTN cell, and like this panel of figure, it is showing you the same mRNA levels but in a different cell lines, and using the vidanolites which showed a better or you can which showed a significant downregulation of mRNA in the first set of screen, right? So TTN and HSC3. So the doses that we have used for the treatment, they are all non-toxic to the cells because we have been using these compounds from quite long now. So for all these cell lines, non-toxic doses have already been like elucidated, and they are using the same. So these all, they do not kill the cell lines as such, right? And not only mRNA levels, we also looked into the protein expression of TMPRSS2. So I'm only showing the data for HSC3, right? So as you can see over here, even for the protein levels, there we could see a decrease in the expression level of TMPRSS2, right? And the same has been shown using like ICC also. Right? Now, what I was talking about in these slides were like pure compounds which have been isolated from ashwagandha, right? But what if we use extracts as such? So Dr. Renu and Dr. Sunil Paul's lab, they are also working on optimization of extraction of vidanolites from these ashwagandha plants. So they have actually seen that using cyclodextrins, most of these vidanolites can be extracted in high concentration, right? So using water-based and like water-soluble and water-insoluble components, so they have created these 4 different extracts from the leaves and the stems. So the first one is the water-soluble like components of the ashwagandha, and the lower one will be water-insoluble components of ashwagandha. So these 4 different extracts shown over here by 10, 11, 12 and 13, and this is the percentage amount that we have used from the extracts that were prepared in the lab. So as you can see over here, these extracts were also able to show similar kind of effect. mRNA levels were also found to be downregulated, and protein expression also went down. And the same thing would be seen in ICC also. So not only pure compounds, but ashwagandha extracts as such were also showing similar kind of results. Right? So this was about the first receptor, like TMPRSS2. This is the, like we also wanted to see what happens to the ACE2 receptor in this, the similar scenarios. So firstly we looked into the residues of ACE2 that are involved in interaction with the RBD domain of spike protein for the docking analysis. So instead of blind docking, we would want to direct it to the structure or the part of the protein in which we are interested. So we created a grid over these residues and performed all the docking studies. I'm not showing the schematic representations over here, but yes, here we used this molecule as a control. So this is also a known inhibitor of like ACE2, and this has also been shown to inhibit H1N1 virus. And this is the docking score for this, and these I'm only showing you the residues which were forming hydrogen bonds. So these 2 residues were involved in hydrogen bond formation. And then if you look at the other vidanolites, a very comparable score, and even some had better docking score than the known inhibitor. And these are the hydrogen bond interactions which were prevalent. And we did similar kind of MD simulations to check the stability of the molecules, and then what, like whether we encounter some change in the interaction pattern or not. Everything was well conserved with respect to the docking studies. And we did similar kind of in vitro analysis, right? So these are the different vidanolites that we looked into. So this panel shows you the like effect on the mRNA levels of ACE2 and this figure shows you the effect on the protein, ACE2 protein, right? Similar ICC images, right? Then the effect of the extract, the 4 extracts on ACE2 mRNA and the protein levels. And this is the concentration of different vidanolites which were found out using HPLC in the 4 extracts that I am talking about. Leaf extracts, water soluble, insoluble, 10, 11, 12, 13 that were shown in those figure panels. And these are the extracts, the extracts prepared from the stem of ashwagandha. So these are the different vidanolites we could find out and what was the concentration present in those extracts. Right? Then, though these were the 2 receptors of the host cells, and we could do the in vitro analysis, but yes, we also looked in only the computational part of it, but we also looked if it can have some effect on the MPRO or the 3CL protease of the viral particle or not. So this is for finding out the catalytically important residues of MPRO. So this MPRO or the main protease of SARS-CoV-2, it is highly conserved among various human and animal coronaviruses. So these are some of those. We did a multiple sequence alignment to look at the residues which are highly conserved and are also known in literature to play an important part in its catalytic activity. So the marked residues are the conserved one and were used for the docking purpose. Right? And here the control molecule used was N3. So this structure is available in PDB as a co-complexed structure. So the same, we just studied the interaction of this N3 with the protein shown over here. So these were all the residues participating in the stable interaction of this small molecule with the protein and was used as reference for comparing our new compounds. Right? So some of the vidanolites that I am showing over here, their docking score and the hydrogen bonds that stayed after the MD simulations. Right? So as you can see over here, this vidanoside 5, it showed much better binding with the MPRO in comparison to others. But unfortunately, because of the being a viral protein, we could not perform the in vitro analysis using this. But yes, then we collaborated with RCB, Regional Center for Biotechnology in Faridabad, to do some viral-based assays. So here we provided them with these pure compounds, and they have taken this remdesivir as one of the inhibitors for the SARS-CoV-2, and they did some in vitro analysis. They co-cultured E6 cells with these different vidanolites and remdesivir as a control, and then they infected it with the SARS-CoV-2 particles, 0.01 multiplicity of infection, and then post 24 hours and 48 hours they collected the supernatant and they looked into the presence of E and N genes, and based on that they calculated these percentage inhibition. As you can see over here, remdesivir, it showed the highest percentage inhibition of virus, that it could not get into the cells, right, somewhere near to 99%. But if you look into the vidanolites, some of the vidanolites actually showed very good percentage inhibition. For example, over here, vidanolite B, right, almost 88% viral inhibition. And then similar to that, vidanoside 5, which also showed very good docking scores for all the 3 different components. Even in the in vitro cell line based assays, this was one of the most potent or potential candidate molecules. And though the range for the inhibition, it is varying for different components. Like it is starting from, I would say, go for this 48 hours. Like if I will say from 25 to even 84%, that is the perfect range. But when you are using extracts as such, you can see that the effect of these extracts could be very promising. I would not say maybe curing the viruses, but at least for the prevention, they could be very good extracts, right? So, similar to ashwagandha, we also looked into honeybee propolis. So, CAPE is one of the bioactive compounds from there. This is the molecular structure, and similar kind of studies, in silico studies, then the non-toxic doses determination, then looking into the mRNA levels, then the protein levels of the same. So, yes, so all these studies also suggest that CAPE also has similar kind of potential and could be very much useful for the management of SARS-CoV-2. And all this work has resulted in these publications. We started with in silico work, and we went on as and when we found more resources and opportunities to perform these in vitro studies, collaborated with RCB. Now recently, like Dr. Sunil and Dr. Renuvarva, they have also signed an MOU with India Institute of Ayurveda, to perform more structured studies and look into the promising potentials and how it can be actually translated and finding the better ways of using it and being more confident about what we have found in the laboratory. So at last, I would like to acknowledge all these people. I have a lot of people to acknowledge. So I'm Mrs. Yazi said I did my PhD from Dr. Sundar's lab in IIT Delhi, and it was that time I got introduced to the this group. People are present over here, Dr. Renu Varma and Dr. Sunil Kaur. So this is my group at AIST, and now I have joined in IIT Delhi. I would also like to thank them because they believed in me in my work, and they have given me an opportunity to now start independently. And yes, the group of Dr. Sudhanshu Vrati in RCB will co-performing all the validatory tests. So that's all. Thank you.
Yes, there. And you mentioned like methamphetamine and methanol and all these compounds, they bind to ACE2 receptor and other receptor. But even mRNA expression is going down when you treat it with methamphetamine. What is the molecular mechanism?
So molecular mechanism is something that we do not know for the mRNA levels, but yes, we do know that it goes down.
Maybe you need to check NF kappa B activity. Maybe ACE2 may be activated by NF kappa B.
So, like we do have a relation of, like we do know that they do have an impact on all these molecules because as I said, we are also looking into the anti-cancer potential. So there we have looked at all these markers, but correlating it with the COVID-19 for now, I cannot directly say because in context of COVID-19 we have not looked at it.
Last year we showed like methamphetamine activates LXR alpha, nuclear receptor, and inhibits the HCG growth. And one of my students, she presented yesterday data about non-alcoholic fatty liver disease through LXR alpha.
So the other reports from the group with the same molecules will have these markers in them.
Okay, thank you.
I think one of the
RCB some antiviral activity data. So have you not checked it with chemostat mesylate?
No, they have used remdesivir as well.
No, chemostat mesylate of course not established as a positive control, but because it is one of the within blood, because in my lab I am not getting any activity with chemostat mesylate. That is one thing, and then second thing is chemostat mesylate already clinical trial was done and it failed in June. It doesn't work in that. So do you think the overall ashwagandha may be working as a good treatment for COVID, but not individual single molecules? Or
as I said, like the individual molecules, they have a wide range of activity from 25% inhibition to 84. So I would not say like pure individual compounds should be used, but extract as such. And we already are working on the optimization of these extracts so that we can have the good yield of these molecules in those. So based on that, we can review. So as market powder that is available, that is prepared using fruits, but our analysis have shown that leaves have the maximum amount of vitamins in that fruits. So probably leaves are a better way of consuming ashwagandha.
the youngest speakers in this conference, young scientists. Very good. Jasprit, I just wanted to know one thing. Do you have any plans to go for in vivo bioefficacy?
In vivo experiments are also done in collaboration with some other universities in the group itself. But yes, we do not have that facility as such at my place for now. Now I have moved from AIST a little. But yes, we do have plans to do that. Yes.
And one thing you said about the propolis.
Yes.
I was expecting some data from that also.
Propolis, actually I just skipped because
I understand.
Yes,
There was a
the data is
limitation of time.
Yes. But it shows inhibition over here as you can see over
Being
here.
an entomologist, I was very much interested to know about that.
So you can see over there, this is the mRNA level and this is the protein level.
It's accepted for publication, so you will have it soon.
So that's why I just skipped it. I could hear the
You mentioned about ACE,
Yes, sir.
angiotensin converting enzyme. Can you imagine whether this ACE is present in arthropods and insects also?
It has some other roles also. If I can relate, it has some cardiovascular related roles as well.
For your kind information, they have been found
So
present
like,
in
could be present.
insects.
So maybe we can look in some other aspects of
Good
it
luck.
as well. Thank you.
Thank you very much. It's great.
Now I request Professor Kumar and Professor